Okay, so far in our first video blog, I talked about resting membrane potential. Then in our second video blog, I talked about graded potentials. And so the resting membrane potential, remember we had said it involved the sodium-potassium pump and leakage channels, and we had said resting membrane potential was minus 70 millivolts. And then we had said, what do neurons do? We had said, well, they conduct a nerve impulse down their axon, and when it gets to the end of their axon, what does it do? It releases a neurotransmitter substance. Okay, so if one of our neurons partners releases a neurotransmitter substance into the synapse where it's communicating with us, which would be what we would call the postsynaptic neuron. So we have two neurons, a presynaptic neuron, one in front of the synapse, and us, the postsynaptic neuron on this side. The neurotransmitter substance would come across, and we had said, what does it open? It opens chemical gated channels. And we had said that membrane potential, it can go all over the place. It can be hyperpolarizing if the neurotransmitter substance that came across opened negative ion chemical gated channels. It would make our postsynaptic neuron more negative. Or it could be, and that would be hyperpolarizing. And we said, that really doesn't do anything. You just have to overcome that, right? We had said, because we have another number that we're going to be trying to get to or thinking about, which was minus 55 millivolts. And so in order to get to minus 55, you have to become more positive, and that's depolarizing, right? And so we had said, if you are a neurotransmitter substance that opens like a sodium gated channel, sodium ion, positive ion gated channel, then you would, instead of becoming hyperpolarizing like the chloride negative ion channels, you would become less negative and move gradually, steadily, maybe, towards minus 55. And we had said that's a magic number. It's called threshold. All right, so if you remember back to the first video blog, I said that these gated channels could be chemical gated or voltage gated. The magic number, that magic voltage that I was talking about, is minus 55. So we have this highly orchestrated series of events that are going to happen when the membrane potential gets to minus 55 threshold. When it gets there, we had said, what is going to happen? Voltage gated channels are going to open. And we're going to have this series of events that is going to be a nerve impulse. And so what we're talking about is action. We're going to have action. This is an action potential. And so a nerve impulse is another way of saying an action potential. Now, when we were looking at the neurons, the cell itself, I pulled out a slide here to show, and unfortunately I don't have a good picture, but if you look carefully right there, it says axon hillock. Usually we show it with a little bump. For some reason there's no bump there, but this is the place where the minus 55 threshold, where the magic happens. It's where all the magic happens, is at the axon hillock. So if you look at him again, it's right there, and here is the axon. So the nerve impulse is going to start here, and it's going to move down to the end of the neuron. And this is where that minus 55 is going to pop open voltage-gated channels. So we've had a lot of different membrane proteins that we've talked about today. Everything from the sodium-potassium pump to the leakage channels to the chemical-gated channels, and now we're adding voltage-gated channels. And so this is a highly orchestrated, it's not like a graded potential at all. It's always exactly the same when we talk about the steps in an action potential. We said the graded potential, it could go all over the place. It could be very hyperpolarized if you've been opening a whole lot of negative channels, or it could rapidly go to action potential if you're opening only positive channels. It's highly variable and goes in either direction. When we all of a sudden result in an action potential, it happens exactly the same every single time. And it's going to start out by opening thousands of sodium-gated channels. So when we talk about an action potential, we have steps in the process. So the very first step is to open sodium-gated channels, a whole lot of them, right there in that trigger zone. So we're thinking right there at that axon hillock that I showed you the picture of. You're going to open up a whole bunch of these voltage-gated channels all at once in response to what? The threshold, minus 55 millivolts. So as soon as you open up all of those sodium-gated channels, you get thousands of sodium ions flowing into that part of the axon. Then the door closes. And so now they're inside. And not only has the potential gone from minus 55, it's gone all the way up to a positive number, like positive 30-ish. So depolarization, you bring all of these sodium ions in. Now, what did we say? We have a whole lot of what on the inside of the neuron? We had said we had that sodium-potassium pump pulling potassium in. So we had all of these sodiums come in, and the door closes. And now you've got a whole bunch of positive charges in here with all of those potassiums. So the second step is for the potassium voltage-gated channels to open, and all of those potassiums go out. Okay, so we've had a flip-flop, and that is called repolarization. The first step, sodium in, is depolarization. The second step is with an R, repolarization, and that's where all of those potassiums leave. And so all of a sudden, we've got all these ions in the wrong place, right? And it happens in that one little spot on the axon. And then you know what it does? It moves down a notch, and you get depolarization, repolarization, and then it moves down a little bit further, and you get sodium's in, potassium's out, depolarization, repolarization, and so on, all the way to the end of the axon. That is that flip-flop of the sodiums and the potassiums. Sodium's now in, potassium's now out. That is a nerve impulse. And when it gets to the end, what does it do? It causes vesicles with neurotransmitter substances in them to release it into the synapse. That is what a nerve impulse is. It's exciting. No, it's not. But it is what it is. So we have all of this sequence of events that makes its way down the axon, so that you go from minus 55 up to about plus 30, and then down to about minus 90. So it becomes really negative now. So it's all mixed up, messed up world. And so you say, well, if that is what a nerve impulse means, then if the sodium's on the inside and the potassium's on the outside, well, you really can't have another impulse until what? What was the first lecture? I mean, the first video blog that I posted. In that very first one, I said you have leakage channels and a pump that's going to be, it's kind of like a leaking boat, and you got the bilge pump pumping water out, right? So in this case, everything's in the wrong place, so you got to pump all those sodiums out and bring all those potassiums back in, and this is going to be what we call the refractory period. All right, so action potential consists of 3 stages. Depolarization. Now you tell me, what happens in depolarization? Correct. All the sodium voltage-gated channels open, and the sodium ions that are outside of the cell in the extracellular fluid, they come flowing in, and the door shuts. Then, next step, second step, repolarization. The door opens on the inside, and all those potassiums go flowing out. Door closes. Then we have refractory period, which is where you start pumping those things, pumping the sodiums out and the potassiums back in. And so the refractory period is a period of time during which an excitable cell, that's a neuron, cannot generate another action potential. And the reason for it, why? Because the ions are in all the wrong locations. Let's see what we have next. All right, one more thing we're going to talk about. I know we've gone a little long here, but it's okay. It's life, right? Okay, so we have this action potential, and so we had said what we're going to do is we're going to compare and contrast that with a graded potential. Action potentials are highly structured. They're all or none. They either happen or they don't happen, and they happen exactly the same way. So it's all or none. It's non-decremental. So what does that mean? It doesn't get weaker or anything. It's the same. It's like, woo, that was a strong action potential that time. No. They're always exactly the same. And they go in only one direction, from the axon hillock to the synapse, to the synaptic knob. That's the way they go. Okay, I'm going to stop right here. We've gone 13 minutes. Yikes! That's a little bit hard for me to upload. That's why it's yikes. Hey, talk to you in a minute.